So first, it's really with a great deal of appreciation and humility that I accept the 2013 Pro Bono Humanum Award of the Galien Foundation. And just very briefly, as I look back on my career in science and in medicine, I truly and sincerely realize how fortunate I have been to be given the opportunity to pursue my passion for scientific discovery in the arena of health. In addition, I've had the added extra privilege as the long-term director of the National Institute of Allergy and Infectious Diseases to be involved in the conceptualization, the support, the enabling, and the implementation of the science of so many other talented scientists with which I've had the opportunity to be associated with it. And so it's really wonderful to get recognition by this group tonight. But as many of us in the room know, and I mean this sincerely, the thrill, the rush of the discovery is really a reward unto itself. But I have to comment that to be presented this award tonight by Elie Wiesel, who has been a long-term one of the true heroes of my generation, and indeed of any generation, is an opportunity, in my mind, is just an opportunity that's beyond my imagination to be here and to receive that. Thank you very much for being here. So I truly appreciate this recognition, and I want to use it as an opportunity to pledge to you that I will continue with all of my energy in the pursuit of the goal that is going to be the topic of my keynote presentation this evening, namely the end of the HIV Aids pandemic. So I will proceed with that, and I hope we can get it on the screen, and there it is. So I'm not going to, it's 8 o'clock at night and we've had cocktails and you have your appetizer in front of you, so I'm not going to show you a bunch of sequences or signal transduction abnormalities associated with immunopathogenesis, what I do. But I want to talk to you about a concept, an aspiration, and what I think is going to be a reality. And it's right here on this slide, namely ending the AIDS pandemic, but importantly, that journey from scientific advances to public health implementation. So before we do that, let's just take a look. Now, there you go, it does work, okay. Let's take a look at the problem we're dealing with. The current status of the global HIV/AIDS pandemic is shown on this slide. There have been 70 million total infections, 36 million deaths, 35 million people living with HIV, and in 2012 alone, there were 1.6 million deaths and 2.3 million new HIV infections. Now, therefore, at the International AIDS Symposium in Washington, D.C. in July of 2012, the theme of the conference was turning the tide together, and Hillary Clinton spoke of an AIDS-free generation. So you might ask, how could we have the temerity, the audacity, with the numbers that I just showed you, to be talking about the possibility of ending HIV/AIDS? Well, I had the privilege and the opportunity to give the opening keynote address at that, and the point that I made, and I hope I made it cogently enough for the world to understand, that all of this could happen, and I say could because we are not there yet, because of the fundamental basic and clinical research that was done from the very first realization that we were dealing with a potential problem. And that basic and clinical research went into the development of interventions in the form of treatment and prevention, which ultimately were implemented, and that allowed us to at least conceptualize and aspire for the end of AIDS. So I want to go through this with you very rapidly. First, let's take a look at the basic and clinical research. I'm not going to give you the details of it, but I do want to show you how much has been accomplished from the very first days when we did not know what we were dealing with, and I was there where we were taking care of patients not knowing what the etiology is. to getting minds in science to do fundamental natural history, and then with the discovery of HIV, to talk about pathogenesis, to develop targets for therapy, and then ultimately to have something we could grab our arms around, namely in the arena of prevention and treatment. This is just one example of that, namely the fundamental basic research of taking the replication cycle of HIV and identifying vulnerable targets. And this really is a shout out to you all in the room, the pharmaceutical industry, to take those fundamental concepts and run with them to the point where today we have over 30 antiretroviral drugs targeting multiple different components of the replication cycle, which have led to a complete transformation in the lives of HIV-infected individuals. And that's what we're talking about when we say the intervention. So when you talk about interventions, let's take the first one, the one that really has been the transforming showstopper of this whole scenario. Now, I show this, some of you may recognize, this is a picture of me in 1982 when we were taking care of individuals who were HIV infected. And as you may have heard from Roy, prior to this, the 10 years I had been involved in developing relatively remission-inducing drugs, and not drugs, I didn't develop the drugs, the pharmaceutical company did, but the use of these drugs to induce remissions in immune-mediated diseases. So I was on a high. All of my patients were getting better. I really felt like I was on the top of the world. For the years that I went from the first patient that I admitted at the NIH in 1981 until therapeutics were developed, I consider them, when I talk about it, the dark years of my medical career, because the mean survival, the median survival of individuals was 6 to 8 months, which means that in 6 to 8 months, 50% of your patients would be dead. Today, with the drugs that were developed by the pharmaceutical industry, with the basic research by NIH grantees, we now, and this is just one study from Hopkins, if a person comes into my clinic, the same building that I showed you on the prior slide, and they're in their 20s, and they're relatively recently infected, and I put them on therapy, I could look them in the eye and honestly say that we could mathematically project that they would live an additional 50 years. That is an extraordinary, accomplishment of that interdigitation with fundamental research and the pharmaceutical industry. Then the other intervention is prevention. There are many of these. We've learned from cold experience that with HIV, you need a combination of prevention. I don't have time and I won't go through all of these, but let me point out from the early days before we even knew what the etiologic agent was, the use of condoms and the use of clean syringes and needles were actually what we call low-tech ways of preventing infection. As the years went by, we had biologically based prevention of mother-to-child transmission, circumcision, and the all-important treatment as prevention. Let me give you a couple of examples of real transforming science. The development of drugs which, when given to mothers during pregnancy and the baby, has essentially taken a 25 to 35% infection rate and had it go down to practically zero. When you look in a city where I live, in Washington, D.C., which actually has an extraordinary prevalence rate, there has not been a single baby born infected with HIV in the last 3 or 4 years. That is extraordinary. Circumcision. We did a clinical trial in Africa, in Kenya, Uganda, and South Africa, and showed that medically supervised male adult circumcision actually resulted in a Are we good? Okay. Actually resulted in about a 55 to 60% diminution in acquisition. The thing I want to point about this that really excites me is that when you look out years after, the results actually get better. So 5 years down the pike, the effectiveness now is about 73%. Would that we had a vaccine this effective. If we did, I'd be talking to you from the Rose Garden of the White House, talking about it with the President of the United States. I'll get to vaccine in a moment. Also, the issue of treatment as prevention. Clearly shown that if you treat early an individual in a serodiscordant relationship, one infected, one not, you treat the infected person, you get a twofer. And the twofer is you save the life of the person you're treating, and you prevent infection in the sexual partner of that individual by bringing the viral load to below detectable level. Now, that's the efficacy. That's the science. Next comes implementing that, which is a huge problem. And there have been some programs, including the President's Emergency Plan for AIDS Relief, which I had the real privilege of being able to put that together for President Bush years ago with the help from a lot of people in the White House. The Global Fund to fight malaria, TB, and AIDS. Philanthropy such as the Bill and Melinda Gates Foundation, Médecins Sans Frontières, and the Clinton Foundation, and others. But importantly, as you're going to be hearing about, it won't work unless the host countries get involved and own their own problem. And that's going to be something that we're actually happening now that we did not see before. So quickly, let's take a look at what happens when you scale up these proven interventions and try to make them go from efficacy to effectiveness. When I went to Africa to put together the program early on in 2002, there were 50,000 people in low and middle income countries who were receiving antiretroviral, usually people who were rich and had access to money and access to drugs. Today, there are about 10 million people on antiretrovirals in low and middle income countries, and there have been about 4 million deaths that have been averted. Importantly, there have been about 1 million babies born HIV-free through the PEPFAR program, something that we would not have imagined years ago. Circumcision actually works in decreasing incidence. So if you go out into the field and you circumcise individuals in areas where they would not normally be circumcised, you can actually reduce HIV incidence by 60%. Put all of that together, and what have we seen over the last several years? About a 30% decrease in AIDS deaths and about a 33% decrease in new infections. So I'm not going to bother you or bore you with the complicated mathematical models, but